there have been many thousands of different approaches to malaria therapy. So that every community in the world that has been subject to malaria infection for any period of time has developed therapeutics that have some efficacy. In terms of Africa, there are thousands of known plants that are used as malaria therapeutics. They don't work to clear an infection. They do work to provide relief from symptoms, so that they are palliative and they are highly valued for this reason. If you think of, for example, if you get a cold and you feel really bad, so colds are viral infections, and we don't have any effective remedies for the cold in the sense that we will eliminate the viral infection. But we do have lots of drugs that you can buy over the counter that will make you feel better, even though they don't clear the infection. So we have a large category of natural drugs that have been used for these purposes. There are other drugs that are now in the Western materia medica that will actually clear an infection and affect a cure. And in fact, the very first drug, disease-specific, was a drug called quinine, or quinine in British English, and that was an alkaloid that could be isolated from the bark of a tree, several species of cinchona tree, or cinchona, as some people say, in South America. And this was the first disease-specific drug, but it would in fact clear the malaria infection. The story of quinine is totally fascinating because for a long time, Europeans learned that it was possible to grind up the bark of the cinchona tree and put it in wine in particular to make a tincture, kind of leach out the alkaloids, and drink it, and you could affect a cure for malaria. And then in the early decades, 1820, of the 19th century, a couple of French pharmacists began to experiment and succeeded in isolating different alkaloids from the bark, such as quinine. And quinine came out of this process as a pure white powder. And this technology, in the beginnings of modern alkaloidal chemistry, spread from France, from Paris, throughout the Western world in very short order. And in fact, this is the beginning of the chemical industry.
There was a race in European countries to find sources to transport the cinchona from Bolivia and other parts of Central and South America, where it existed, into groves. And they developed plantations in Indonesia where the trees grew well and were selected for high levels of quinine in the bark, and has led to the synthetic aspects of drug development. And so now we have a whole plethora of drugs which can be used for combination therapy, which is what's sort of evolved now in the 21st century for treating malaria.
When we think about the intensity of malaria infections in Africa, which is higher than anywhere else in the world, and where almost all the infections were falciparum, which is extremely dangerous. This helps to explain why Europeans, in the course of imperial expansion, colonized North America, Central America, South America, South Asia, Southeast Asia, parts of the China coast, but not sub-Saharan Africa. It was considered too dangerous for Europeans because of the malaria burden. And the colonization of Africa by Europeans lagged centuries behind the colonization, for example, of the Americas or of South Asia. And parts of West Africa were known as white man's grave for this reason. So it was only in the course of the 19th century that Europeans embarked upon, in the 1880s, what was known as the scramble for Africa, and where Europeans came to colonize more than 90% of the continent in very short order. So this is another kind of interesting dimension of malaria history, in that it shaped in a most fundamental way the experience of Africans during the 17th, 18th, and well into the 19th centuries. At the same time that the Atlantic slave trade was transporting Africans into the New World, African states in the continent itself, below the Sahara, were largely independent of any type of European rule or influence. Another point that I want to make that goes back to our earlier discussion on cinchona is that the quest to produce large amounts of quinine had substantial economic and environmental impacts. Again, the reason why it was so important to have plantations of cinchona was because then you could strip the bark from the trees, ship the bark to factories in the United States or in Europe. But the bark could be ground up, the alkaloids could be isolated, and then they could be bottled and sold. The malarial burden in the tropical world in particular was so heavy that it was the principal challenge of European empire. European states were trying to rule non-European peoples. They needed some military presence. They needed an administrative presence. There were also missionaries. There were commercial agents. And they bore this very heavy burden. And so to lighten the burden, the Europeans concentrated on the production of plantations of cinchona trees. They tried them everywhere. They tried them in California. They tried them in Algeria. They tried them in India. They tried them in Sri Lanka. They tried them a lot of places around the world, but only in a few areas were they successful. And only in a few areas did they realize, only belatedly, that they had planted out the wrong kinds of cinchona trees that had relatively low percentage of alkaloids in them. In Sri Lanka, formerly Ceylon, the island off of the southern coast of India, European planters, British planters, deforested much of the highlands of Sri Lanka in the largest process of tropical deforestation in British Empire. And they planted out at its height in the ground and in pots getting ready to be put out into the fields. They had to give you an idea of the scale, they had 120 million trees planted in the highlands of Sri Lanka. These were massive undertakings. And they were massive undertakings because they were trying to address the principal medical problem of European empire in the tropics. As Clive was mentioning, Indonesia, they succeeded in producing high quality cinchona trees because they addressed a scientific program, or developed a scientific program of tree breeding. So that they took a type of cinchona tree known as Cinchona legeriana, which had a naturally high amount of quinine in the bark, and then they planted out trees and sampled each individual tree, and then destroyed all but the best bearing trees, and then planted them out and went through several generations of this, sampling every individual tree and destroying all but the best. And eventually they produced a super species of super trees that were capable of producing large amounts of quinine, and the Dutch dominated the market and developed a cartel in quinine that was based in the Netherlands. And they pretty much controlled the market for quinine globally. However, during the early years of the Second World War, when the Japanese took control of the Dutch East Indies, the European access to quinine was cut off. And so this produced initially quite disastrous consequences for the Allies. Allied troops in the Pacific Theater were put out of service, either killed or injured by malaria in relationship to Japanese bullets on a ratio of 10 to 1. The troops were literally decimated, and Europeans for a while had to withdraw and regroup in the Pacific Theater. This is what launched yet another very important phase in the American chemical industry, which was the search for a synthetic quinine that could replace the natural quinine that was no longer available. And eventually, chemists came up with a number of different drugs that were highly efficacious. And as Nina was mentioning, the spectacular success really was that of a drug called chloroquine, which was so inexpensive and was produced by many different nations that it successfully reduced malarial infections in Africa at least by half, and succeeded for a couple of decades until the widespread use eventually selected for parasite resistance. And there became then another crisis in Africa beginning in the 1980s when chloroquine was no longer effective, and yet at that point we didn't have any next generation drugs that could really fill the gap.
It's interesting you say this because I followed this personally in Zimbabwe, in Rhodesia as it was. We used it extensively for treatment, even more than should be, because if you look at the situation in the field, the diagnosis of a malaria case required drawing blood, making a slide, sending the slide to a center where experts would look at them, and then it would take weeks before the clinic actually got a response. So they used chloroquine extensively. Anybody who came in with a fever, with anything, a headache, whatever, they were treated with chloroquine. So chloroquine used extensively for things that they should never have used it for. But it was, and it's a very good example of the overuse of important drugs.
Clive brings up a really interesting point about the problems of the diagnosis of malaria, because it often presents with symptoms that could be something else, kind of like COVID today. But Clive has been the director of the Johns Hopkins Malaria Center in Zambia, and he was really a pioneer, the pioneer, in developing a new diagnostic technique called rapid diagnosis. And so would you say a little bit about that, Clive? This is the man.
I had moved to the United States and I was working on malaria, and a student came to me and she said, She works for a diagnostic company, and they have a test, a rapid diagnostic test for malaria. So I said, I really don't believe that, but it was actually developed in Baltimore in a lab very close to where I lived. So I visited the lab, and it was demonstrated to me, and I thought, this is absolutely wonderful. So they gave, at that stage, I was working in Bagamoyo, in Tanzania. So they gave me a carton of this test. We took it into the field, and we tested people. And literally within 10 minutes of taking a finger prick of blood, you could have a positive diagnosis or a negative diagnosis. And I think that this was actually the biggest step, the most important step. And I remember defending this concept in meetings in various parts of the world, that I felt that this should be done, and that it should be developed. And my colleagues in the field also felt this, but it took about 20 years before it became available. And so beginning in the 21st century, the distribution of the rapid diagnostic test was extensive. It's still, to me, the most important diagnostic we've got, because even with PCR and that, you can't get a reading in the clinic immediately.
I have personal experience with Clive's testing. So I got dengue a few years back after coming back from Cameroon, and I was lucky that Clive brought me into his office and tested me right there to make sure I didn't have malaria. But also, I wanted to, as a scientist myself, I wanted to highlight how amazing to have this test is, because when you're in the field in Africa and trying to do diagnostics, here there aren't many problems with doing a simple PCR test. Polymerase chain reaction. It's a little machine that you put on top of your desk. It runs within a couple of hours. You get results. It's very easy, here in the States. If you go to these little villages in parts of Africa, for example, where I've done some field work, you are often experiencing power outages. The ranges in temperature varies a lot, which really impacts your tests. You also have to be able to train technicians to be able to run these tests. And you also have to be able to pay them to be able to come in and do it. And sometimes the volume of tests coming into the clinic is just so large you can't keep up with the scale that's happening. So it's actually a really big barrier to understand the full burden of the disease when you can't even just test for it. So now that there's available, just a little stick, just like the COVID test that we have now, where you put a little drop of blood or anything on it. There are also people who are trying to develop saliva tests to make it even easier, that you just spit on something to see if you have it. That's in development, but it's getting easier and easier to look for this stuff right on the spot. So it's really revolutionizing data collection and being able to know the right treatment.
Recently, there's been a lot of work, but also publications on a vaccine for malaria. And the vaccine that we have requires a single shot, and then a month later another shot, and then a month later another shot, and then a month later another shot, and then 3 months later. So there's replicated treatment is required. In the experimental work, this can be done because there are huge resources to make it sure that when a person is taken into the study, that they actually get these serial treatments. But if you're working in the field, this is very difficult, because the clinic doesn't have transport to go out to the houses, so the people have to come in. people lose track of time. They also have other commitments, and they can't be sure to have the second and third and fourth doses. So the use of a vaccine is experimentally feasible, but very, very difficult to implement. Very difficult.
And this is the concept in public health that's called vaccine hesitancy. So that just means that you're not sure if you should get this vaccine or not. And it's because of all the reasons that we're talking about, that as a person, let's say again, living in that village in Cameroon, and I have to go 2 miles walking to get to the clinic, I might actually really want that vaccine and be like, I really want to be protected. I want my kids to be protected. But is it worth it for me to travel 2 miles? Because what does that also mean? I will miss out on work. Because most likely you will not have time off from work to go there. I have to do something with my kids. Am I going to bring my kids with me 2 miles walking? Once I get there, I might be worried about, is it going to cost me something? Because you might not have internet to go, or a telephone to go call someone and say, and so that's actually a big factor, is how much is this going to cost me? And also how much money am I going to lose from doing that? And these are really important factors to think about, not just that, in any of the treatment plans that are out there, is, how easy is it for people to go get that? And what are those specific barriers, and how do we overcome them? And it's a huge challenge in these spots.
If we think back to the middle of the 20th century, at the end of the Second World War, when the early years of the World Health Organization, the principal disease problem in the world seemed to be malaria. And while it was known that there were these different parasites, principally vivax and falciparum, and lots of different mosquito vectors, it seemed possible to launch an assault across ecological zones on all species using a new chemical weapon. And that this weapon was so effective that it might be possible to, in fact, eradicate malaria. So not just to deal with therapeutics and the problems of quinine and then chloroquine and other drugs that were coming along, but to kind of definitively end the transmission of malaria. And to do that, the approach that was adopted was mosquito control, and specifically the use of a new type of chemical insecticide. And I'm wondering if Clive can say a little bit about this, because Clive was active in these campaigns and knows a great deal about it.
The concept of eradication actually goes back to the middle of the 20th century, where WHO decided that we were going to eradicate malaria. And there was a program, and I actually worked for, in my country, the Federal Malaria Eradication Program. So it was set up like this in the 1960s. By the 1970s, it was obvious that this was not going to work, because there was a major study done in Nigeria by WHO, and 3 or 4 different European research groups to see if they could eradicate malaria from a district in Nigeria. It didn't work because we didn't understand the biology of the parasite, the extensive diversity of the mosquito population, so it was left as a future policy. So eradication can be thought of, but it's extremely difficult to implement, because it actually means eradication. It means globally eliminating the malaria parasite. We can't eliminate the mosquitoes. We can't.
Well, Clive, what was that miracle insecticide?
DDT.
DDT.
Yes. Well, they were actually, we are still using DDT actually now on small small control programs because of its impact on the biotome that we live in. We've got to be careful about the use of these things.
Well, DDT has a really interesting history itself. And correct me if I'm remembering the details wrong, but it was first widely used in World War II. And it's just so effective against so many different kinds of life and insects. And it was used, I remember watching videos of, soldiers, for example, and they'd get a big shoot of DDT over their heads because it was an effective lice killer, for example, and that was a big problem in the army. But then it was used after that agriculturally, and so there are lots of old images of airplanes going over crops and dusting the crops to get rid of the nuisance pests and things like that. And then it was found to be effective as an infectious disease controller as well. And actually, this is how I got into all of this research, is because way back in the 8th grade, I read Rachel Carson's Silent Spring, which was about against the indiscriminate use, so this widespread use of DDT in the world around us. And it was the first time that someone had published that, hey, there's actually some negative consequences of this pesticide. So as a young girl, I was just very moved by this work, and so I was actually anti-pesticide for a while, until I started doing this public health work, and you saw how miraculous it was at controlling the disease in certain parts. However, what I appreciate in current times is that people are really against the indiscriminate use, this going up in a plane and just dumping it. So now it seems really effective is that you take a spray canister and you go inside a home, for example, or in very specific spots where the mosquitoes are breeding, and you spray it right there. So it's a spot treatment. It's not like you're just going around and fuming a whole place. So this seems to be a pretty effective method.
I think this brings up this really core issue of trade-offs. I mean, it is not going to be effective in bringing about the eradication of the disease. And that's something that got established really during the first efforts, 1955 to 1969, in the global malaria eradication campaign of the World Health Organization. And it seems also to be the case that the repeated use of DDT or some of the other synthetic insecticides has diminishing utility.
Yes.
So the question is, well, what is the role of residual insecticides in malaria control? To my knowledge, and correct me if I'm wrong or if you just have a different take on this, but I think that there's general agreement that it's appropriate to use these types of insecticides when you have an expansion of a malaria zone into an area where people do not have any acquired immunities, and in order to prevent an epidemic, that it's extremely effective in a single use or multiple uses, but not ongoing, to shut down epidemic processes. In some other areas, and I think this is true like in the low veld between Zimbabwe and Mozambique, you have a program of repeated use of residual insecticides simply to control, with the understanding that there will be diminished efficacy, but with the idea that it may be possible to rotate different classes of insecticides and to slow down and prevent.
My perspective is that you've got to have a good health infrastructure operating at the clinic level so that the population is kept alert and helped and treated as appropriately by trained personnel. This is how we will control malaria. And this is actually being done in certain parts of the world, but there are plenty of other places where there just is not a good health infrastructure working at that level. So the importance here really is, how do you ensure that the clinic nurses are properly paid, are kept alert, are given the equipment and the encouragement to maintain a program. And this is how we will keep it under control, is public health.
Hi, everyone. To everyone in our audience, and on behalf of our project director, Professor Stephen Bednarski, thank you for all coming today to the second event in our online symposium series, Disciplines and Dialogue. If you'd like to ask questions, make sure you've clicked the link to watch it on YouTube or Facebook so you can add questions into the chat. And I'd like to extend a very special thank you to our speakers, Drs. Clive Schiff, Jim Webb, and Nina Martin, for an insightful and thought-provoking discussion. The video that you just watched was edited by our excellent communications team here at the Dragon Lab, Gillian Wagoner, John Loudfoot, and team lead Aaron Kurian, who directed the video and spearheaded the editing process. The generous funding and support for this symposium series comes from the Social Sciences and Humanities Research Council of Canada, St. Jerome's University, and the University of Waterloo. Joining us now for our question and answer period is Jim Webb, Professor Emeritus of History at Colby College, who conducts research on the history of disease. He is the founding editor of the Ohio University Press's Perspectives on Global Health and the series In Ecology and History. He has been awarded grants from the Wellcome Trust, the U.S. National Institute of Health, and the Institute for Advanced Study at Princeton. He has held recent fellowships at the Rachel Carson Center in Munich, the Center for the Global History of Development at the University of Shanghai, and is a Fulbright professor at the University of Botswana. His recent books from Cambridge University Press include The Guts of the Matter, A Global History of Human Waste and Infectious Intestinal Diseases, The Long Struggle Against Malaria in Tropical Africa, and Humanity's Burden, A Global History of Malaria. Thank you for being here with us, Jim.
You're welcome. Thank you for doing such a nice job on the film. It came out really beautifully.
I'm glad you like it. Yeah, that's our team's put a lot of effort into it, and yeah, it's really come along nicely. So to start off with the questions, we have one from the audience. Dr. Webb, how did you first get involved in the study of malaria?
That is a great question. I first got involved in the study of malaria when I was carrying out research on quinine. And I thought I might write a book on the history of the first disease-specific drug in the Western Materia Medica. And I talked to an editor and friend at Cambridge University Press who said, "You don't want to write a book on quinine? You want to write a global history of malaria." And I thought, no, I don't. I don't think I'm really the person to do that. I don't really think I have the training. So I then just kind of began to educate myself in the scientific literatures about malaria. And I thought that, yes, it might be possible to do this in a truly kind of multidisciplinary way, and to take account of the historical literatures and the biological literatures and the biomedical literatures. So that was really kind of my first extended foray into this subfield of historical epidemiology.
That's very interesting. And along those lines, how have you seen the field change over the past decade or since that book? And where do you see it going in the next 10 years or so?
I think that it is small and growing. There are a number of younger historians who have gotten training at the master's level in public health and sometimes in other of the biological sciences, and that this is allowing for more scientifically informed investigations of past disease processes. So I would say a lot of this research has been taking place in Africa, and that it's not exclusively the case there, but I mean, there's been a lot that's been done and is continuing to be done. And I think and hope that this field of historical epidemiology will gain an even broader reach.
And what do you think are the most critical challenges facing malaria prevention efforts today?
This is a great question, a central one. I think that it's a complicated one because there are many components to successful malaria control. One of them is the organization and carry through by political authorities. And so that malaria control is in some ways dependent upon successful governance. And in many states around the world, we have successful governance, but in others we don't. So it's extremely difficult to prosecute a program of malaria control without that. Another dimension of this is simply access to financial resources. So some financial resources can be garnered from within the state. Some have been on the basis of grants or of loans. And some of the international donors have, with enthusiasm, committed large sums to malaria control or a malaria elimination, which is kind of a shorthand for talking about malaria eradication in certain zones that could then be extended. So there's been a lot of money put into malaria elimination, but this has also been subject to broad global financial forces. So during economic downturns, the commitment of donors and the commitments of governments that had committed money have waned. And so the funds that some governments have counted on for malaria control have been significantly reduced. Then we also have a problem just at the level of the functioning of public health departments. So as Clive was saying in the film, when you have public health departments that don't have reach into some of the areas within a national territory, it's very difficult to diagnose malarial infections. It's very difficult to treat them, and it's very difficult to intervene in the case of major outbreaks or epidemics. So one of the challenges isn't specific to malaria control itself, but it's kind of embedded in this larger question of, well, how do you staff, how do you fund, how do you maintain robust public health infrastructure in states that are struggling with political problems or struggling with economic problems?
Well, that's very interesting. And that gets me thinking, if we're thinking about historical comparisons, the successful banning or limiting of DDT, for example, I just wonder if you wouldn't mind elaborating on how that worked so successfully. To what extent was government impetus the driving factor? How important was the publication of Rachel Carson's book, Silent Spring? Can you just give us a little overview of that?
Sure. There are several parts of this question. One is the efficacy of DDT itself. DDT was highly efficacious among, in many areas, and against many of the important mosquito vectors, but not all. And so that there were other residual insecticides that were used in the campaigns of the World Health Organization and that are used today. And some, such as dieldrin and BHC, benzene hexachloride. So it wasn't exclusively a matter of using DDT. DDT, when it was used, in some populations of mosquitoes, produced resistance, sometimes pretty quickly. So it was a very, very powerful intervention, but it was not one that was universally successful. Rachel Carson's book, Silent Spring, in 1962, was certainly an eye-opener for a lot of people. And it resulted in the banning of DDT in the United States for agricultural purposes. Many people do not realize that Rachel Carson herself was not opposed to the use of DDT in public health and for disease control. She thought it ought to be used, with extreme circumspection, but it was not something that she was against, kind of as a matter of principle. So DDT, during the era of the global malaria eradication campaign and thereafter, was principally used for agricultural purposes. And I remember as a child in the American South, watching dusting planes go over the cotton fields, and myself being, covered with DDT as a result of getting close to it. So the movement away from the use of DDT had somewhat ironic effects in that in areas in where it was banned for agricultural purposes, some farmers moved to the use of other insecticides, and some of which were more dangerous than DDT. So there was a very kind of unintended consequence to the banning of DDT. There were certainly environmental activists who were opposed to any use of DDT for disease control or any other for public health purposes. And an effort took place around late 1990s to try and ban the use of persistent organic insecticides. And it was only when African states organized themselves and objected to this initiative, claiming that it was necessary to have exceptions to this rule because they realized the importance of continuing to use DDT or other residual insecticides for disease control. So as it is now, we have a lot of areas in the world where, in fact, farmers are still using DDT on a large scale. And then we have in public health the rather circumscribed use, largely in sub-Saharan Africa, not even all throughout sub-Saharan Africa, but just in some areas.
Very interesting, and challenges some of our ideas about even how Rachel Carson saw DDT and all that. It's very interesting. Mm-hmm. We have a question from the audience. Is the history of malaria and how we treat the disease unique, or can malaria and its treatment provide an instructive paradigm to help us tackle other diseases? And if so, how?
Wow, tough question. I would say that many disease phenomena have some similarities. If we take malaria, which is a vector-borne disease, so it's dependent upon the mosquito. So that's pretty different from respiratory diseases, for example, like COVID-19, or many other infectious diseases that are spread, through fecal-oral transmission or other means. So within the world of vector-borne disease, there are certain common challenges. I mean, if we consider, for example, probably the second largest vector-borne disease challenge in the world after malaria, dengue fever. Dengue has a different vector. It's not one of the Anopheles species, but it's an Aedes mosquito, or a couple of different species, major species of Aedes mosquitoes. Then the same kinds of questions are right there in the forefront. How should we control mosquitoes? And should we use residual insecticides? And what is the role of, for example, personal repellents and screening on houses and the like? But within the world of vector-borne disease, the behavior, for example, of the mosquito-borne diseases is dependent upon the behavior of the mosquitoes themselves. So some mosquitoes are very happy taking blood meals from animals other than human beings, and they don't cause any problem. There are some mosquitoes that really are focused on human beings, particularly in Africa, and really prefer blood meals from human beings and are almost uniquely focused on human beings, which is one reason why the malaria burden in Africa is so high. And then you have other mosquitoes that will happily take a blood meal from a cow or a bird or a dog, as well as a human being. And where these animals live, the mosquitoes, whether they breed in domestic circumstances, like around houses. This is the case for the Aedes mosquitoes that carry dengue. So they breed in flower pots, they breed in abandoned pails and Coke cans and lids that are in people's yards. They breed domestically. And so the ways of going after those breeding areas are different than the kind of breeding areas of most of the malaria vectors. So it's a complicated matter. There are some common issues, but the biology of the mosquitoes themselves, as well as the pathogens that they carry, is really kind of the linchpin that determines the types of interventions that might be effective.
Very interesting. And we have a question from a multi-part question from Tim Newfield about basically the role of the historian with malaria today. And it is, what role can historians play in curbing malaria incidence and prevalence today? Has it been a struggle to get historians to use their toolkits and evidence to improve healthscapes today? Or how can we encourage historians of disease to engage more with the present?
Great question, Tim. Thank you. Complicated. There's three of them. I'm not sure. Maybe you can give them to me again, one at a time.
Yeah. What role can historians play in curbing malaria incidence and prevalence today?
This is a great question. I believe that research on the history of malaria control can play a significant role in informing contemporary policy. This is because most people who are involved in malaria control are trained in the biological sciences or in biomedicine and are unaware of the history of malaria control. So that the general upshot of this has been that in malaria control, some of the same ideas and the same tools that were used decades ago are being used again without any awareness of what the successes were and what the problems were in the past. So there's kind of a disconnect between contemporary public health practitioners and policymakers and what is known by historians about what happened. I remember giving a talk to the malaria group at the American Centers for Disease Control. And I was talking about the insecticides that were used during the first campaign in a few areas in West Africa. And that there was a lot known about how effective they were against the different mosquito vectors, some pretty successful, some not. And the response from the CDC was, this is so interesting. This is exactly the experience we're having now. They're using the same insecticides in the same areas against the same vectors without understanding that this had been done previously and what the consequences were. So I think that bringing this kind of evidence forward in shows like this, or in speaking to people in policymaking positions and practicing groups is important. Second question.
That's great. Well, and the second question basically is the other side of that. Has it been a struggle to get historians to use their toolkits and evidence to improve healthscapes today? And how can we encourage them to do that?
I think we can encourage historians to do this, but I think it's really only a subset of historians that is going to be interested, because in order to do this, you have to kind of be interested and learn about the various scientific aspects of interventions. So you have to be interested in epidemiology. For malaria, you have to be interested in parasitology. You have to be interested in insecticides. And because most historians come out of training in the humanities, or sometimes in the social sciences, rather than the sciences, I think this is going to continue to be a challenge until we start producing young historians who have broader training.
And so when you teach this subject, what are the approaches that you use to teach it to students, and what takeaways do you hope that they have from this subject?
Well, I give them pretty challenging materials to read, because even though I taught in a department of history, I would say that most of my students in advanced seminars were not history students, but were in biomedicine or in biology or in global studies, and typically had a broader background than historians, the young historians that were being trained.
Based on some of these answers, I can maybe guess what you might say, but a question we like to ask in these seminars sometimes is, what question are you most tired of hearing about the subject? And what would you say so that you never had to hear that question again?
My gosh, I'm not really sure that I would answer that in a way that you might think. I don't tire of questions about historical epidemiology or about malaria or vectors or insecticides. For some reason or another, I find the material fascinating, and I'm always happy to talk to people about any and all issues that have to do with malariology, to be honest.
Fair enough. That's great. Yeah. Are there any popular misconceptions that you wish you could give one explanation and just dispel? What's the number one misconception, would you say?
About malaria?
Yeah.
Well, I think that in the Western world, there is a general lack of awareness about the role of malaria in the Americas, for example. And most Americans are not aware that malaria was a serious problem throughout the 19th century and well into the 20th century, and that it had an important role in the peopling of the Americas and in the movement of Euro-American and African populations. It's certainly not, a single the single important explanatory factor, but it's definitely kind of an integral part of the peopling of the Americas. And for those in your Canadian audience, it's probably somewhat surprising if I were to say that, there used to be malaria in Canada.
That does surprise me, actually. So yeah, it's very interesting. The area that our project studies in southern England had malaria at one time too, which people don't usually think.
No.
Has the COVID-19 pandemic, and it's sort of a topical question now, but has the COVID-19 pandemic had an effect on malaria prevention efforts or misconceptions about the disease, or has it had any effect in that way?
I think that the demands to counter COVID-19 have absorbed some monies that might otherwise have been allocated to malaria control. And I think that, I guess that's what I would say.
And we're almost out of time, but I'd like to ask, what are you working on now? What's next for Professor Jim Webb?
Thank you for that question. I am interested in exploring with colleagues in the microbiological sciences the impact of micronutrient deficiencies in diet on metabolic disorders. So that there are some diseases, for example, the major disease in the American South in the first half of the 20th century was a disease that's known as pellagra, in which is a vitamin B3 disease, niacin, and deficiency as well of one of the amino acids, tryptophan, that was very, very widespread and killed about 100,000 people. And we had around 3 million clinical cases and almost certainly 10 million or more subclinical cases. So there's some evidence that suggests that this type of stress in micronutrient deficiencies, and this is true of others such as beriberi, which is another vitamin B deficiency, as well as outright malnutrition, can have effects that are passed on epigenetically to succeeding generations. So I'm interested in exploring with others the possibility that the historical impact of micronutrient deficiencies and malnutrition may have a significant explanatory value in understanding, for example, understanding the epidemic of type 2 diabetes, for example.
Well, that's about all the time we have here today. Thank you very much. Thank you again to all our speakers, and especially to Professor Webb for kindly answering our many questions. If you have any further questions, please don't hesitate to reach out to our team here at dragonlab.ca or medieval-environment.com. If you'd like to sign up for our mailing list, we send frequent updates and notifications of upcoming events. Goodbye for now, and take care.
Thank you.